Hi, everybody. So, since today was kind of a disaster in terms of internet stability, and this is kind of a complicated topic, I decided to record what I may or may not have said during class today so that you can refer back to it, because I know that we didn't get to a lot of what I intended. So, here we are at Elephants Day 6. We're starting this CSI Wildlife Lab. And today and Friday, we're going to be working on case 1, and we will move into case 2 as soon as we're done with case 1. It's all the same interactive module. I've just split it up into 2 separate worksheets to make it a little bit more organized. All right, so the first thing you want to do is click on this CSI Wildlife link, and you will come to a page that looks like this. To launch the interactive modules, you click on this green button that says Launch Interactive, and a brand new window is going to open where everything is going to be available to you. The videos, the quiz questions, the information, the maps, everything that you need to complete the worksheet for case 1 and also for case 2 is all included here. All right, so the other piece that you're going to want to take a look at is the Google Doc called CSI Wildlife Case 1, which has all the questions that you need to answer. The module, the interactive module, has all the information. The Google Doc is where you're going to be recording your answers. And so here is our case 1 Google Doc that we went through in class. And you just want to read through the questions and it'll tell you what to do next. So the first one says, read the introduction and watch the opening video. And what we are listening for is, what is a keystone species? We'll come back to that. And then, what is the percent of African elephants killed each year? And what I said in class is that it's really important that you be able to calculate the percent of a whole. It's useful in everyday life for many reasons, but in particular in being able to get a sense of, somebody throws out a number, 50,000, and seems like a big number, but is it really a big number? You don't really know until you compare it against the overall total number. And so getting a percentage of the whole is a nice way to be able to say, in relationship to all of it, is 50,000, is that a big number or is it a small number? We just don't know until we get into it. All right, so then we're going to go back to our interactive thing. We're going to play our video.
Elephants are the biggest land mammal. They are what we call a keystone species. And by keystone species, it means that when you pull that species out of the ecosystem, the whole thing starts to unravel.
Most elephants live in protected reserves throughout the African continent, yet a great number are illegally hunted each year.
Right now, we estimate that there's 50,000 elephants a year being killed, and there's probably less than 470,000 left in Africa.
Poachers kill elephants for their tusks, which are sold primarily to make ivory objects. When law enforcement officials seize a shipment of ivory, Sam Wasser works with them to trace where the elephants were poached.
We just analyze large ivory seizures. Those are seizures that are at least a half a ton in weight and are worth upwards of $1 million or more.
He applies forensic tools based on DNA analysis to pinpoint the elephant population that the ivory came from.
We wanted to know, where was the large organized crime syndicates operating in Africa? We need to figure out, where can we target law enforcement so that we can shut this trade down? And we have a really urgent problem, and we really need to get a handle on this.
Okay, so that's the video, and we're going to move on and we're going to solve our first case, which is case 1. So we click over to the next area, but we have a couple of questions to answer before we move on. So we go back to our worksheet. What is a keystone species? Well, just like we talked about with the sea otters, Sea otters and wolves are also keystone species, and what we talked about in class is that when there were not enough sea otters, the sea urchins were able to become overpopulated, and they cut down all of the kelp forests in their area, which threw the entire ecosystem out of balance. And the same thing happened with the wolves in Yosemite, in Yellowstone. When the wolves were not there, the herbivores, the deer, variety of different kinds of deer, were allowed to overpopulate, and the vegetation disappeared. We had soil erosion, the rivers changed course. It threw the entire ecosystem out of balance. So a keystone species is a species that if you remove it from the ecosystem, what happens is that the ecosystem gets thrown out of balance. Okay. And it kind of throws things into chaos. Everything falls apart, is I think what he said in the video. Okay, so that's the answer to this first part here. The second part, we're calculating the percent of the whole. So he said that 50,000 elephants per year are killed, and there's only about 470,000. So if we do the math, I've given you a hint here that you want to divide, in order to get the percent of the whole, you divide the total number of elephants killed, which is 50,000, you divide that by the total number of elephants, period. And so what you get is, I'll just write it here, so 50,000 divided by, which we represent with our / 47,000 equals, I think we said it's 0.106. That's what we came up with. And when you multiply that by 100, what that gives you is the percentage, which ends up being 10.6%. So 10.6% of all elephants are killed every year because of poaching. That's a pretty big number. So now when you see the percentage, it gives us a good idea of, is that a big number or a small number? 10% of all elephants in the continent of Africa, that's a big number. Okay, so now we're moving on to case 1. And it's telling us to watch the crime scene video on the case 1 slide, and then talk about the goal, the overall goal of this project. So we're going to watch this video right here. It's an embedded video, and there's information down here which actually tells you exactly what the goal is. All right, but we're going to watch this little video so you can have them tell you. Okay, so we've learned from the video that they have confiscated tusks that were found in the back of a truck, and they've sent them to a forensic lab, CSI lab, CSI Africa, to try and figure out where did these tusks come from. The place where the poachers were caught was here in the Congo, and there are 3 possible parks that surround the Congo that all could have been where those elephant tusks were gotten. And speaking of the goal, the whole goal of this is to be able to target law enforcement. We talked about how one of the ways to stop poaching is to make sure that there are plenty of park rangers and police officers out looking for poachers. But you know, the continent of Africa is huge. There's just not enough manpower to have sufficient coverage for all of these countries and all of these areas where poaching might be taking place. So by using forensic evidence to find out exactly where did these elephant tusks come from, it allows our higher ups, our political people, our police enforcement to target where do we need to send our people. We can't send them everywhere, they'd be spread too thin. But if we know exactly where these tusks are coming from, we can focus in on those areas and stop the poaching in those areas. Okay? And then one of the questions is to look at this map and to pick out 5 countries that elephants live in. And all you have to do is look for the areas in green, and you just have to read the map and figure out what are 5 countries where that you see some of these shaded green areas. So there's plenty of things to choose from. There's Tanzania, Kenya, South Sudan, the Congo, Angola, Cameroon, all over the place. Just pick your 5 favorite countries. See if you can discover some new countries you didn't know existed before. Okay, so we've talked about the goal. We've talked about the 5 countries that elephants live. Now we're going to start to talk about DNA profiling. And what I did in class is I said, okay, the first thing we're going to do is we're going to learn about gel electrophoresis. So I've included 2 different videos that I'd like for you to watch. We watched the Amoeba Sisters one in class, and it talked about the properties of gel electrophoresis. That DNA is a negatively charged molecule, and when you put it on a gel, the beginning of the gel is negatively charged. The end of the gel is positively charged. So the DNA is going to travel towards the positive charge. The large pieces of DNA are going to move through that gel much more slowly than the small pieces of DNA, which are going to move very fast. Okay? So when you apply that current to the gel, the DNA separates according to size. The small pieces moving fast, and they end up farther away from where they started. The large pieces moving more slowly, and they stay closer to the top of that gel. Okay? If you want to learn more about gel electrophoresis, I've included a video virtual lab where you can walk through the process of setting up your own gel electrophoresis. If we were together in class, we would be doing exactly that. But since we don't have that opportunity, I've put a video here for you. We're also going to be talking about PCR, the polymerase chain reaction. So I've included a video here that explains it, I think, a little bit better than the one that's embedded in our module. Okay, so going back to our module here, we're going to move on and start talking about how DNA profiling works. Okay, so we're on case 1. We've done the crime scene. Now we're moving on to how DNA profiling works. Okay, so here's an example of a gel electrophoresis. We've got a DNA ladder, which gives us DNA bands, which are of known size. So you'll notice the size here is measured in base pairs. So those are the bases along the strand of DNA. 200 base pairs is the longest one we have here. And I can't read it because my thing's in the way, but I think it's 50 would be the lowest number. So you'll notice that the smaller pieces move farther away from the starting point. This gel electrophoresis gives you an opportunity to create a DNA fingerprint for different individuals. Same thing we would do at a crime scene if we were collecting blood or body fluids of other types, or hair, or tissue left behind, right? We can use that DNA to identify suspects, and we do it in exactly the same way as what we're showing you here, except this time we're applying it to elephants, and we're trying to figure out which of these elephants do these tusks belong to. Okay, so it's a forensic technique, and we're introducing this idea of STRs. One of the questions is, what does that stand for? Well, it stands for short tandem repeats. And you'll notice in this strand, this example strand of DNA, you can see that there's a sequence that repeats over and over. CTAC, TAC, 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 so it's a TAC that repeats over and over and over again. Sometimes it's 3 base pairs, sometimes it's 4 base pairs, right? But it occurs within DNA all over the place, and when we target that piece, the short tandem repeats, the areas on the sides, the flanking sequences, right? So the nucleotides adjacent to those repeats are called flanking sequences. The alleles, so we talked about having different alleles. Well, the alleles are going to be slightly different sizes. So if you look at a gel, no two elephants are going to have exactly the same DNA pattern if you're looking at multiples of these short tandem repeats, because there's going to be mutations. All right, so let's move back and look at some of our questions so we can kind of frame what we're doing here a little bit better. Okay, so what does STR stand for? Short tandem repeats. It tells you right there in the module. What do the bands on the gel represent? Well, the bands correspond to pieces of DNA of specific lengths, right? And it's not just one strand. One strand of DNA is too small to see. You have to be able to amplify it. We're going to talk about that in just a second. So the next question here says DNA fingerprinting. So getting that pattern of a DNA fingerprint on a gel like that is similar to actual fingerprints that you would collect at a crime scene. All right. So this question is asking, how is DNA fingerprinting similar to actual fingerprinting? And what we said in class is that both of these are very unique for an individual. So just like no two human beings have exactly the same fingerprint pattern, no two human beings, and also elephants, no two elephants have exactly the same DNA fingerprint. Every one of us has a very unique DNA pattern because of all the hundreds of thousands of genes that we have. They're never all identical. Okay, so our DNA fingerprint is just as unique as our actual fingerprint. Now, how they're different is that a fingerprint is visible. I can see a fingerprint. It's easy to say, yep, there's a fingerprint. I don't need any special equipment. I can just look at it. A DNA fingerprint, by contrast, is at the molecular level. It's microscopic. I need special equipment in order to be able to visualize a DNA fingerprint. So that's how the two of these are different. All right. And then the module goes on to talk about how are DNA samples from elephants collected. And they gave you a bunch of ideas for different sources of DNA. Okay, they told you on the next slide, you click to the next, and it will tell you here that you can get DNA from a lot of places, including hair, saliva, body tissues, tusks, and even dung, which is a fancy word for poop. Okay, so you can get DNA from all these different places, but most of it is in a very small amount. You can imagine if I only have a single hair, and all I have are a few cells, a few skin cells that are attached to the root of that hair. That's not enough for me to run on a gel like this. So I need to amplify that DNA in order to make more of it so that I can begin to work with it. And so the polymerase chain reaction is one way that we can make more of very small quantities of DNA so that we can begin to see it on this gel. And how the electrophoresis works, if you click this start button, you'll see exactly what you would see if we were running this for real. So everything started at the top, and over time, the larger pieces moved slowly, the smaller pieces moved very quickly, and so you end up getting a very nice separation of different DNA bands according to size. Okay, so watch that PCR, the polymerase chain reaction video, to get a better idea of how we go from a single strand, a single double helix of DNA, and we end up with hundreds of thousands of copies of that piece of DNA so that we can actually begin to work with it. Okay, so now we're moving on to the application part here, and we didn't really get to this in class, so I'm going to stop here and I will create another one of these videos for moving on into the application area. This is where we left off, but let me just look and see for the questions here. So we watched the video on the polymerase chain reaction, and we said that those flanking sequences are important because that's where our primers are going to bind, and it's going to start to amplify, replicate the DNA that's in between. So it's amplifying those repetitive sequences, and it's very easy for then us to visualize different alleles across different elephants. But it's those flanking sequences that allow us to make multiple copies of just that DNA that's in between them. Okay? All right. Question 9. So we've got a particular STR fragment. There's a particular allele that we want to take a look at. And we make these primers, and we're looking for 10 different elephants. We're looking at 10 different elephants. Would you expect those fragments to be the same size? Would each one of those 10 elephants have exactly the same size DNA at that particular location? And the answer is no, they wouldn't all be the same size because DNA mutates. If you look at 10 people, some of them will have exactly the same gene at exactly the same place. But the chances that 10 human beings or 10 elephants would have exactly the same allele in one area of DNA is very unlikely. There's many different alleles, and mutations happen all the time, so we would not expect 10 elephants to show exactly the same pattern, even if we're only looking at one particular location. All right. Question 10 is talking about the PCR reaction itself. And it's kind of like a soup. I can put in multiple of those flanking sequences, and I can begin to amplify all different kinds of those tandem repeats. It doesn't have to be one at a time. I can do 10 at a time. I can do 17 at a time. And what makes the biggest difference is those flanking sequences provide a uniqueness that allows the reaction to multiply many different genes at the same time. So it's all about the flanking sequences. That's the secret here to number 10. More about gel electrophoresis for question 11. What is the relationship between the size of a DNA fragment and the distance that it migrates? Well, the larger DNA fragments are going to travel slower as it moves through the gel. The smaller the DNA fragment, the faster it's going to move through the gel. Okay? Now, why does the DNA migrate to the positive electrode? Well, it's because DNA molecules have a negative charge and are therefore attracted to the positive electrode. It's not really like a magnet. That's the way electricity flows. It's always negative to positive. And so negative molecules will be drawn towards the positive electrode. Okay? Then it says run the gel in the technique section by pressing the start button. We did that. And we were looking for what is the largest and the smallest fragment. Okay, so let me just back up and I'll show you that piece. I'm going to back up to technique here. And when we ran this gel, one of these elephants had both the largest and also the smallest DNA piece, and that would be this guy over here on the right. So this is the largest DNA piece, band, because it didn't move, it moved the least amount. So it's the largest, and you can look over here to find out how many base pairs this corresponds to. My markers here are between 200 and 180, so if you guesstimate about 190 base pairs, that would be roughly correct. The smallest piece of DNA is way down here, and it is somewhere between 60 and 80, not exactly half, so we might want to call that like 65. All right? So it's the elephant on the right that has both the largest and the smallest, and the approximate size is 190 for the largest and 65 base pairs for the smallest. Okay? Now, we're going to get to this next part on Friday, so I will make you another video, maybe even before then, so you have it in advance. All right.